Okay, Life Systems Labbies, this is the Photosynthesis Lab Part 2. I've got the screencast kind of on the sidebar here, or I'm sorry, the net meeting menu on the sidebar here, so just bear with me if I have to answer some of the questions here on the side. So we've got the key concepts in this. We're looking at electron transfer happening while photosynthesis occurs. So we're trying to measure photosynthesis within the leaves, and as photosynthesis occurs, electrons are freed up, and they will be absorbed by, in this experiment, a pigment called DPIP. So you've got pigments, chlorophyll. We've looked at the different kinds of pigments involved encompassed in leaves. You've got the thylakoid membrane. This is kind of breaking down the anatomy of the chloroplast. Let me go ahead and see if I can get that to say. So as the photosynthesis occurs, electrons are freed up, and then they will be absorbed by this pigment called DPIP. It's like a what would you call it? Like a dye that's going to become less and less blue, directly correlated with the rate of photosynthesis in the experiment. So as the leaves, I believe it's like spinach leaves, let's pretend it's like fresh spinach leaves, are being put under conditions that would make them photosynthesize, that DPIP is going to go ahead and absorb those electrons and become less and less blue. So I'm going to go ahead and replay that. So in the experimental tube, we'll have these spinach leaves put in an environment where they will be conducting photosynthesis. And I wonder if, looks like it doesn't want to replay it like it's supposed to. So let me just go ahead and, for some reason it's just not working to replay it. So when the light strikes the chloroplast, the DPIP will be reduced and go from like a dark blue to a lighter blue as photosynthesis carries on. So we've got, for a device that we use in this experiment, a spectrophotometer. This is just an instrument that shines light through a tube and will give you the percent of light absorbed. So in this slide, it's basically saying we're going to take the tubes and zero out our instrument, make sure that it's reading properly, and then we'll use our experimental tube. So we have like a blank or a control tube, and then we make sure once that control tube is reading the way it's supposed to, then we start to use the experimental tubes. This is all kind of preliminary setup stuff. Is everything clear at this point, Jill? Okay, good. All right, so now when we set up the experiment, we're going to have 4 tubes that are going to have, I believe it's just water, and then there will be like you take like a hole puncher and just punch out pieces of like spinach leaves or really fresh green leafy something, and you'll suspend them in the water, and then you'll put DPIP pigment or dye indicator in each of the tubes, except this initial one. This initial tube 1 is not going to have any of that blue pigment, that blue dye. These other 3 will have dye, water, and spinach leaves, and we're going to have unboiled spinach leaves in the tube 2. We'll have boiled spinach leaves in tube 3, but then we're also going to cover it in foil, so this will be basically a tube that's mimicking darkness. And then your DPIP boiled and subjected to light in tube 4. So we have these 4 tubes with varying conditions: boiled and unboiled chloroplast, no pigment, no light, and we're going to put them through this experimental setup where we've got a light transmitting. Now this will be light that would induce photosynthesis, and then we have this big water bath that'll kind of distribute that light out, so it's not like gonna cause heat to actually affect the experiment. This water bath is meant to sort of absorb any heat that might happen from the light. So in an oxidized state, as these chloroplasts in the spinach leaves photosynthesize, they will turn the blue dye in these tubes lighter and lighter to the point where they'll be almost completely transparent. So, um, so this is how the experiment is set up. Are there any questions about that, Jill? Okay, excellent. So every 5 minutes, we will actually take these tubes then and we will put them in the spectrophotometer and measure how much light is actually being absorbed by the blue pigment, the DPIP dye in these tubes. So you'll have the blank tube, which will be the control tube to make sure that your instrument is reading properly. And then there are some helpful hints here. Let's see. First reading provides a baseline. Keep the test tubes clean. Make sure that you use a Kim wipe so that there isn't any finger smudges that would alter the readings. Mix the sample before you measure. Be sure all the tubes in the spec are in the same orientation, so there's not any variations in the glass that would mask your results. So like if there's a label on your tubes, you don't want to have that. And then maintain a constant distance from the light source. You want to keep them all kind of equidistant from the light source. Good. Oops. All right, so this page is really just going to be like a cut and paste into a Word document. You'll go ahead and copy and paste. And then as if you would be taking measurements every 5, 10, and 15 minutes, you would actually mark down the percent light transmittance that you get, the reading that you get from that spectrophotometer. So there's really nothing you can record on this sheet, but I would copy and paste it into a Word document, which I think I have right here. And then you'll sort of suppose, I think it's asking you to hypothesize what sort of graph you would see in each of the tubes. So this tube 2 is the one with the dye and the unboiled chloroplasts exposed to light. This second graph will be what you suppose the percent transmittance over time you'll see in the DPIP, the one with dye, unboiled chloroplasts, and in the dark, so it's got the foil around it. And then this third graph, tube 4, will be the DPIP, boiled chloroplasts, and light as well. So it's up to you then to photocopy to copy this into a Word document and actually write down on these graphs, kind of trace a line that shows how much transmittance you'll see in those tubes over time. That means that we start with a very dark tube. The DPIP will be a very dark pigment, so you'll start with very low transmittance of light through the tubes that have DPIP in them. And then you have to decide under those conditions that these tubes are subjected to whether that blue dye is going to, whether photosynthesis will increase, and then that blue dye will become lighter and lighter over time. Does that make sense? Are there any questions at this time? Okay, so the question is, what is the purpose of the blue dye? So let me go ahead and see if I can keep this in there. But then, it looks like I'm not going to be able to answer any questions. Hold on, without losing my screen. The purpose of the blue dye would be to, if we go back to this original concept, you can see that the DPIP is going to be blue, and as it's called an electron acceptor, so as photosynthesis occurs, electrons are freed up and absorbed by that blue dye. So again, I'm going to repeat that because it's an important statement. As photosynthesis occurs, the light comes down, photosynthesis occurs in the chloroplast, electrons are freed up and then absorbed by that blue dye, and they become lighter in the process. So we're basically using this indicator dye as a measure of photosynthesis occurring. As the photosynthesis increases, the rate of photosynthesis increases, the blue dye becomes lighter and lighter. So another student question, wouldn't the wouldn't the dye become darker because they're absorbing more? Well, so you have to think in terms of what they're absorbing. If they're absorbing electrons, the electrons aren't really pigment per se. They're just, it's just energy. And so as these electrons, which are colorless, are absorbed, it doesn't really impact it doesn't really add to the darkness of the pigment dpip. It actually makes the dpip appear lighter. So it's really like a biochemical reaction happening. Okay, so let me go ahead and see if I can catch up with where we are. It's kind of the flaw of these labs is you lose track of where you were. Okay, so you're really just going to be tracing and bringing in, you can submit it in hard copy. If you were to do it in Word, I guess what I would do is you could insert a shape and then like pick an arrow. I would maybe make the arrow a different color, but you can draw your line in here either electronically or you can, I wonder if I think I can make this a different line, but you can either electronically insert your curves or your lines however you see. You've got different curvy, wavy ones if you want, or you can just bring it in hard copy. Go ahead and draw it on your graph and bring it in that way. So you've got a couple different options on how you want to submit this. That's the other reason why I extended the due date for this lab. Normally it would be due electronically to the Dropbox tonight, but I've extended the due date to next week, so you can go ahead and bring it in hard copy without any penalty or late turn-in. So then you've got some questions. Now this lab quiz is modifiable. You can go ahead and try to answer the questions, and then as you submit them, we're running out of time for this screencast, so I'm going to go ahead and show you that you can go out and come back in. If we go back to this, and you can retry the questions. And then again, you'll cut and paste those into your Word doc as well. So that final document is what you'll submit electronically or bring in to class next week. Thanks and peace out.